Okay, it is my distinct honor to introduce this year's Lee Hartwell Lecture, Dr. Susan Gasser. The Lee Hartwell Lecture is given by a noted researcher in the field who has used yeast in a way that has obvious impact on other fields. And as you will hear in Susan's talk, and as I will describe in a minute, Susan has definitely had major impact or profound impact on many different fields. Susan began using yeast as a graduate student. Already then she had creative and interesting ways in trying to have a system for protein import into mitochondria using yeast as a source for material. Her interest in chromosome biology began when she moved as a postdoc to the Lemley lab, but in that lab she used cell cultures and Drosophila. She must have realized that yeast are way cooler because when she started her own lab, she switched back to yeast. When she started to work on, it was in 1986, at the Institute for Experimental Cancer Research, and she used yeast to study various aspects of chromosome biology. And over the years, Susan has made important contributions in many fields of chromosome biology, including chromosome organization, telomere positioning, gene silencing, DNA replication, the response to DNA damage, DNA repair, genome stability, and I could go on, but I was running out of space on my slide. And so perhaps more than anything else, Susan's studies made us appreciate the contribution of chromosome intranuclear localization to the importance of chromosome function in all of the areas that I've just described before. And the quintessential image that comes to mind when you think of Susan's work are these three concentric circles with a dot of where the chromosome is. And using this simple system, Susan initially showed that chromosomes in yeast have distinct locations, and the locations of the chromosomes are very important for chromosome function. And using this and other assays, Susan went on to find proteins and processes that contribute to chromatin dynamics and localization within the nucleus, and most importantly, how these affect chromosome function. In the past decade, Susan expanded her work to C. elegans, where she's studying, among other things, the impact of chromosome nuclear localization on development. So as you can imagine, over the years, Susan has won many awards and has been recognized as a leader in the field. She's been elected to the Academy of Sciences in numerous European countries. She's served on many advisory boards, international committees, and she's received more honors than I have time to list right now. But I would like to note two honors in particular. First is the EMBO FEBS Women in Science Award, and the second is the Weizmann Institute Women in Science Award, both of which are given, and I quote, to women who've been inspiring role models, which Susan obviously has. So with this, I'll turn the podium over to Susan.
So it's a great honor. I thank Lars for swapping with me since I couldn't stay Saturday night. So it is a distinct honor to get the Lee Hartwell Award because Lee has always been one of my heroes. And I won't talk about the cell cycle. In fact, I've never truly worked on the cell cycle. Lee, but as a graduate student, in my first introduction to genetics, yeast genetics, was when Jeff Schatz carefully went through the 1974 Lee Hartwell science paper, and it was my first realization what epistasis was, what terminal phenotypes, conditional mutants, all this was new to me. And I've religiously made all my students read this paper with me because I think it's beautiful. Lee had a great conceptual drive behind his science, and it's something I've always striven for. So I'll be talking about the role of chromatin dynamics in nuclear organization in genome integrity. Actually, there's one other thing I wanted to say. Lee Hartwell should not have only gotten one Nobel Prize for the cell cycle. He should have gotten one, or maybe he still will, for checkpoints. And checkpoints at least are relevant to what I'm going to talk about. So, but what Lee used to analyze cell cycle mutants was a combination of microscopy and genetics. And basically, that's what I do. I combine microscopy with genetics, whether it's C. elegans or yeast. and try to understand biological processes. And what's changed is that biology is no longer a 2 or 3D problem. It's about spatial organization over time. And that lets you understand processes in terms of rate-limiting steps. And that's exactly what you get when you get a terminal phenotype at a particular temperature. That's a rate-limiting step, that gene that you knocked out. And where genetics does it, we're trying to understand the same rate-limiting steps in biology. So my space-time team is these 3 fellows. Vincent's left the lab, has his own lab now, but Michael and Andrew are both graduate students. So what we've been doing is studying chromatin. That's been actually the theme of my laboratory ever since it was established. And studying space-time relationships has led us to study DNA damage and repair. So our genomes are mutated or have lesions, thousands of lesions per day, 40 to 50,000 per mammalian cell per day. Most of them are rapidly repaired. They are adducts, they're single nicks, etc., and very few double-strand breaks. But we all fixate on double-strand breaks because if you have a double-strand break, you can lose a chromosome arm, and they provoke translocations that are, of course, cancer-causing. Now, damage doesn't occur on the naked template on which most of the repair processes occur and have been studied. It occurs in chromatin, and you can have damage as frequently in compact heterochromatin as in open chromatin. And so it's actually the chromatin context in which we have to study the cellular events of DNA repair. So so my theme has been how does chromatin respond to genomic insult? So in budding yeast, as in most species, there are 2 major pathways of double-strand break repair. There's NHEJ, which enjoining, which can be error-free. It's most rapid. It's the most dominant one in most species, and in G1. And then homologous recombination, which is extremely efficient in yeast, but which requires resection. And usually the repair is from the sister, which is not damaged normally in normal. That's not true in our systems, but it's true in life. Now, if you have a double-strand break and are not repairing from your sister, then you have to search for homology. And budding yeast not only repairs efficiently off the sister, but it repairs efficiently off ectopic donors. And so this notion of homology search is something you can study in yeast. And if you have a double-strand break and a resected end, and you have to find a template, then homology search is most likely the limiting step. At least there have been theoretical papers that propose that, and some experimental paper from Jim Haber's lab. So in order to do homology search, you're going to have movement either of the damaged template or both. Now, movement is one of those things, chromatin damage movement is one of those aspects that you might say was discovered in yeast, or at least we elaborated it in yeast, and it turns out to be relevant for many, many other species. I don't see the mouse. Do you see it? No. Could you put the mouse on? Huh? Ah, there it is. Okay, so this is taken from a review from Megan King, actually. But what we have shown in budding yeast is that persistent double-strand breaks that cannot be repaired by end-joining or sister homolog repair actually get shifted from whatever location they are in the nucleus to associate with the nuclear pore, or in some cases in S phase, with the sun domain protein called MPS 3. This helps in some way processing that end for an alternative pathway of repair. Now it's turned out that this is true in many other species. You can see the references down there. And in Drosophila, double-strand breaks in heterochromatin have to translocate out of the heterochromatic domain in order to be resected or at least to bind RAD51, and then actually also transfer to the nuclear pore. And this mechanism turns out to be very conserved. Also, shortened telomeres, which in budding yeast associate with nuclear pores. Same thing, well, at least in mammalian cells, an uncapped telomere is shown to actually increase its movement and then also be influenced by dynamic movements of the sun domain proteins of the nuclear envelope. So these things are conserved. And a very recent recap of data in this field shows that actually three papers published just in the last year show, again from Irene Ciolo, that the double-strand break in heterochromatin in flies and the persistent double-strand break in yeast and the eroded telomere actually all have a similar pathway that controls where they end up, and that involves assimilation by, in budding yeast, the cis1-2, or the MPS3, MMS21, and then cis2. And if the proteins at the break get polysumolated, they bind a stubble, which is a sumo-dependent ubiquitin ligase, and get shifted to the nuclear pore. And if they're only monosumolated, they seem to associate with this sun domain protein. And the outcomes of repair from each pathway are different. So this is, again, a conserved mechanism, and more likely than not, it's also going to be true in mammals. But what I really wanted to talk about is not just where things end up, but what allows chromatin to reposition itself, whether it's directed motion, random walk, motor or cytoskeleton dependent, etc. And this is what we started roughly almost 20 years ago. But as soon as Andrew Belmont and Aaron Straight came up with these LAC-O arrays, I realized that we could actually look at the telomeres and the origins and everything that we loved in vivo and track them. And so we combined it. This was our contribution to the field, the NUP49 GFP, because that gave us a way to actually do very careful microscopy and localize the position of a spot with relationship to the center of the nucleus, so we could use haploid yeast and lots of mutants to study position and dynamics. And what happened was that we realized the dots are not, stationary. They move quite a bit. We did 3D tracking over time, usually in Z stacks, and then project them usually because there's too much data for 3D deconvolution, and then analyze it biophysically by calculating the distances moved over different time intervals, and then you plot that in what's called a mean squared displacement plot, where the slope here is proportional to the diffusion coefficient in the plateau, and you'll always get a plateau in a nucleus because that shows that you're not diffusing to infinite space, but within a confined domain. But actually what turns out is that it diffuses within much smaller space than the whole nuclear diameter, and these constrained domains of diffusion are quite reproducible. They're roughly 0.6 microns in radius, and that is about one-third the volume of the nucleus. So many years of analysis of this kind of movement told us that it depends on where the tag is. If you're near a centromere or telomere, you're going to move less. It depends on the ATP levels, the growth of the cell. It depends on the phase of the cell cycle, and there's much less movement in S phase because the two sisters are held together by cohesin. And there's also a matter of local chromatin context. But what we really wanted to know was whether there was any use for this movement, and one of the obvious things might have been that double-strand breaks have to find, when both sisters are cleaved, you have to go on a search for your homologous donor, and that this might, in fact, involve looking for sequence homology outside that radius of constraint that the sequence usually moves in. So we induced a double-strand break. This is the system. We just put an ICSE site on chromosome 14 with a tag. We wanted to get away from MAT because the chromosome 3 is very special, folded up. And so we then tracked the RAD52 spot after cleavage. And in S phase, we're cleaving both sisters, and in G1, we're cleaving one. And in both cases, this is the mean squared displacement plot, you see that the mobility that you normally get for a locus is increased roughly fourfold, from about 13% to 47% of the nucleus. This is less than the 0.6 microns of radius. It's 0.46, but that's because it's an S phase and not G1. And it's really important in all these studies to separate S and G1. So the question was, you know, does all damage move a lot? And the answer is no. You can have Rad52 foci that don't move, and it depends on whether they can be repaired from the sister or not. And a flip nick, for example, doesn't increase movement. It's a Rad52 focus. It's processed, but it doesn't increase movement because it only occurs when the sister is next to it, and it's a single-strand lesion. So seeing that this might be a controlled, reaction, we asked whether the checkpoint was involved. And indeed, if you cause a double-strand break in a MEK1 or a Rad9 mutant, you see that this increase in mobility doesn't occur. So it is a controlled event. And we did the next experiment was to ask, do you actually need the damage? Can you just activate the checkpoint and increase mobility? And amazingly, this works. So we use the David Toksisky method where you just co-localize DDC1 and DDC2 and activate MEK1. Here you see the activation by co-localization of DDC1 and DDC2. And you ask, does activation of MEK1 alone increase mobility? And indeed it does. So again, a mean squared displacement curve. Not as much as a double-strand break, and we know that processing and the repair proteins at the double-strand break contribute to it, but nonetheless, checkpoint somehow signals to the nucleus it has to move more. It's a controlled reaction that the checkpoint response stimulates movement. It also requires resection and a decision that NHEJ and a homologous repair off the sister isn't going to work. So you may think of chromatin mobility as enabling alternative types of repair. So the question then arose, is that increase in mobility at the site of the break, is that only happening at the site of the break? Checkpoint is signaling to the whole cell, basically. And so we decided to track another locus. And that was all stimulated because Rodney's lab and mine published back-to-back. And he mapped a movement at a non-damaged site, and we had only mapped locus breakage sites. I wasn't sure I believed his results, to tell you the truth, and so I had to do it myself. And I mean, to tell you the truth, I don't believe anybody's results, so don't take that personally, Rodney, unless I've done it. I'm a super skeptic. So we induced damage either with double-strand breaks or with zeaxanthin. And indeed, we tagged a locus. We looked at multiple loci. And we had Rad52 in the cell, fluorescent, so we knew that Rad52 wasn't binding. We weren't looking at damage, a damaged site and its increase. And indeed, we see that with increasing damage in the nucleus and activation of the checkpoint, movement goes up. Again, it's dependent on the MEK1 Rad53 activation and the INO80 remodeler. Now, what about INO80? This brings us back to my love, chromatin, right? So we're talking about something moving chromatin in the nucleus, and so I was really pleased when we checked mutants in these two remodelers, both of which are recruited to double-strand breaks, and saw that actually both of them affect the dynamics of the double-strand break, but only INO80 actually affected the movement in trans. Even more exciting was when other studies in my lab and the lab of Ashby Morrison showed that many of the subunits of INO80 are targets of MEK1 or Rad53. And so that really piqued my interest, and we continued to study knocking out this ARP8, which is, so in many of the cells we study, INO80 is an essential gene, but its essential cofactors like ARP8 and IES4 and IES5, you can knock out, and lose activity of the remodeler. So if you knock out those and induce a double-strand break, these two bands here that don't show any difference are ARP8 delta cut and uncut, and you see there's no increase in movement, whereas the wild type one goes up. Now, INO80 also has actin subunits, and the whole field who studies movement is fixated, and with good reason, on microtubules and actin. If you knock out microtubules, you actually get more movement. So microtubules are more of an anchor than, more of an anchor than the force. But because the spindle pole body and MPS III can link to the actin cytoskeleton, and because the nucleus itself is rotating quite a bit, people wondered whether actin filaments were involved. So we depolymerized actin with LAT-A, and in the absence of damage, this is a different way to portray dynamics, you see that there's less movement. That's without damage. But interestingly, it didn't increase the diameter within it moved. It just increased some kind of decreased aberrant movement. So actin cytoskeleton is acting on LAT-A, but when we did damage, we could still see an increase. This is the wild type damaged and cut site, and the LAT-A cut site. You still see an increase in dynamics, so it's not like the absence of the actin cytoskeleton eliminates the increase in movement. And if you look at an ARP8 mutant, so this INO80 mutant, plus and minus LAT-A, you got exactly the same. So either, if this says that LAT-A and ARP8 and INO80 are epistatic for movement. Okay, so let me just recap. What I told you is that undamaged locus moves in a limited domain. When it's a cleaved locus that has no sister or end joining possibility, it moves more. This depends on INO80, MEC1, and RAD51 at the cleavage site. And then at undamaged loci in a cell that incurs low damage, you again see that chromatin generally is moving more. Again, it's MEC1 and RAD53 dependent, INO80 remodeler dependent, but not HR repair factors. It still occurs, the increase in dynamics still occurs in the absence of actin filaments, although actin cytoskeleton does alter nuclear rotation. And so we were wondering what happens to chromatin. So, So this is all unpublished. The question really is, if we're seeing a change in mobility, and DNA is a chromatin fiber that is constrained by its polymer fiber characteristics, then we might expect that there might be changes generally throughout the nucleus in response to damage, and this might be actually responsible for the physical movement that we're observing. So the first thing that came to mind was histone modifications, of course, and on our way to look at histone modifications, we were doing very, very careful quantitation of histone recovery. So my student, Mickey, would take wild-type cultures, treat them 1 hour with damage, either zeaxanthin for an hour or gamma radiation, and leave them a few minutes, and then harvest them to do careful quantitation. And of course, we do, he has very nice Western blots, optimized everything. And repeatedly, he could see that histone H3 and H4, and also H2A and H2B, actually reduced their levels, both on zeaxanthin, here it's quantified from mini blots, and on gamma-IR. Of course, he showed me this. I told him, that's impossible. Yeast doesn't lose histones. You can't live without your full complement of histones. And in fact, he's right. We did SILAC very, very quantitatively and isolated the chromatin afterwards and did mass spec. And again, all of the core histones lose about 20% of their composition, but not HTZ1. I still didn't believe it. I told you I don't believe things. And so we did it fluorescently and took a tagged H3 CFP and actually watched it in real time and compared with a whole slew of other proteins to see if there's selective loss after an exposure to zeaxanthin. And this is the time course of HTZ1, but the same thing is true for NUP49 or DDC2 or anything you want to look at. They don't go down. But H3, sure enough, loses it, and already within 30 minutes, you've lost about 20% of your H3. So is the proteasome involved in this? So we inhibited the proteasome and did the same experiment. Here you see by Western blot the H3 going down in the presence of zeaxanthin, the absence of an inhibitor of the proteasome, but it at least is less down in the presence of proteasome inhibitor. And these inhibitors are not 100%, so I actually believe it's a proteolytic degradation. Finally, we wanted to do the mapping of nucleosomes to know if there are just a few spots that are losing all these histones and then other spots that are not. And together with Tom Owenhughes, we did nucleosome mapping genome-wide in the presence and absence of zeaxanthin. Now, it's very difficult. The positioning is easy to do, but the quantitative occupation of sites is not so easy, so he spiked in non-Cerevizi chromatin and did the mapping. And sure enough, in the absence of nucleus of damage and in the presence, what you see is a global loss of histones of about 17%. And it didn't matter where you were. High transcribed gene, low transcribed gene, you see it at the +1 nucleosomes, you see it in the promoters. There seems to be a pretty general loss of histones. So again, it's a controlled reaction. It happens here in wild type, and you don't need the repair machinery, but it's blocked when you interfere with the with the checkpoint MEK1 or RAD53 or in 080. So again, it's the same process. Now, in the last 2 or 3 minutes, I want to say what happens when you lose histones. You don't die. This is reversible. But we did more and more sophisticated analysis of chromatin dynamics by doing the microscopes get better. We can do it much faster. We do stacks now in 80 or even 10 milliseconds when we visit Genelia farms. But anyway, it gives you much, much more beautiful data. But it also allowed us to work with a physicist that could model the polymer using a beta polymer Rouse model, and show that he would predict that actually we should be seeing, by his, decondensation and a loss of constraining forces, as you increase diffusion rates and lose confinement. So we asked the question, do we see decondensation of the polymer fiber? And so we did this just by taking a domain of about 10 to 20 kb that was fluorescently tagged and measure the volume on hundreds of spots. And indeed, in damage, we see that chromatin expands. This is what you would expect, basically, if you lose histones, because histones package chromatin, of course, into smaller domains. We tracked individual loci along chromosomes to ask, do the chromosomes expand? That was just one locus expanding. If you take different colored spots along the chromosome arm, do we get systematically larger distances? And in fact, here's, you see an increase. The pink is the damaged, or here the red is the damaged. Both on chromosome 14 and chromosome 3, you see this expansion of chromosome. Finally, you don't want to always work in the conditions of damage, so we downregulated histones by putting them under a Gauss control. And here you see you can shut down histone synthesis, and by 30 minutes you're down to about 70% of the histone complement. And again, we see that chromatin decompacts and mobility increases. So this is, you don't really need damage. You don't have to have the checkpoint if you can get rid of histones in another way. So the final question is, does it impact repair? Of course, that's what everybody wants to know. And so we did a strand invasion assay by qPCR. And indeed, if you impair increased movement with a RAD9 mutant, you do delay strand invasion events for an ectopic recombination event. It's not true at MAT because of chromosome 3 being folded up, but it's true if it has to search, if a double-strand break has to search in the volume of the nucleus. So this is what we've shown, that actually the cell responds to damage in a very remarkable way. If you cannot repair by end joining or sister repair, the whole histone complement gets at least partially affected, and chromatin itself expands, perhaps with the idea that expansion will allow access to for repair machinery. The question, of course, is this conserved? The phenomenon is true. Mobility increases upon damage, as several labs have shown, even in mammalian cells and definitely in flies. Whether the mechanism is the same, I don't know. And that's where we are today. So I'd like to thank my lab, always a great lab. This work has been done by, again, Michael Hauer, Andrew Zieber, and Vincent Dion. Thank you. And I don't know if there's questions. Yes. There are questions.
Quick question. You said H2AZ levels are not going down?
Yes.
So, well, obviously H2AZ is part of a nucleosome. So does the +1 nucleosome not change and all the rest do?
No, actually we do see some of the +1 nucleosome going down. But we know that HTZ is specifically deposited at damage, so maybe it's being shifted from some promoters to some damage. But that one doesn't go down.
Hi, thank you for the talk. So I wondered if you ever looked at the susceptibility of specific regions in the genome to break, not to repair, but to break, and if so, does it correlate to nucleosome occupancy?
Oh boy. I know that people have looked at breaks under conditions of replicative stress on HU, and those are these hotspots of damage, and they tend to be at highly transcribed genes. But I think there has been a mapping of breaks, like how random gamma radiation modifies, and it seems to be random. It doesn't seem to have preference. And this has been true even in organisms that have much more compact chromatin and much more open, extremes between compact and open chromatin. So I don't think that, if you're talking about environmentally caused damage, no. If you're talking about, there should be no difference. If you're talking about mechanistically, internal mechanism caused damage like replication, fork transcription, collision, then, it could well be.
Okay, thank you.
Thank you.